Hello friends, welcome to my channel Sims Biology for a new video. In this video lecture, we are going to discuss those two terms that are very important, linkage and recombination. So we will discuss how recombination is resulted due to crossing over and how recombination helps in appearance of non-parental characters in the offspring and how linkage helps in bringing the parental characters to the offsprings. So let's discuss in detail about these two terms. So first, as I have already told you in my previous lecture, that I will discuss with you about two different scenarios. So let's see what was the first scenario. So I am writing here scenario 1. So what happened here in the first scenario, we will talk about when the genes are located in two different chromosomes, how the characters are transferred to the offspring. So let's talk about, we are talking here about two different characters. Okay, first let me write here. So we will have big flowers. So flower character, we are taking the characters of flower. What type of flowers are found in the plants? So flower, we are talking about big. For big we have capital B because it is the dominant character, and likewise for small size of the flower we have small b. Likewise for the color, this is the for the shape or size, yes, size of the flower. Next we will talk about the color. So we are taking the color as the second character of the flower. Here for red we have capital R, and for white we have small r. Okay, so these are the two different characters we are taking for discussing their linkage and recombination. So in scenario 1, I have already told you, we will discuss that when the genes are located in two different chromosomes. So genes are in two different chromosomes. So what will happen here? Let's discuss about this. So you can note down that we are taking two different characters, the size of the flower and color of the flower. Now let's discuss when the genes are in two different chromosomes, what will happen. So I can write down here, this one is a chromosome having the gene capital B that represents the big size of the flower. Likewise we have another chromosome where the capital R is there representing the red flower. Okay, likewise we have the small b representing the small size of the flower and small r representing the white color of the flower. So when we fertilize these two plants, so in the first case we have capital B, capital R, and we will fertilize this one with another plant having small b, small r. Okay, so I can denote here capital B. This one is also capital B in two different chromosomes, capital B. Likewise here we have capital R and again capital R. Okay, likewise in this case we will have small b, small r. This gene is here in one chromosome and this one is in another chromosome. So this is the condition. When we will cross between these two, what will be the probability of gamete? Capital B and capital R. Here we have small b and small r. Okay, by crossing these two we will get the offspring capital B, small b, capital R, small r. Okay, so this will be the product. So when we cross this F1 product, this one is F1, this one is P1, it's G1. In F1 we have a hybrid plant having the big size of the flower and red in color, because big and red are dominant over small and white. Now we will cross the F1 with double recessive, means we are going to do the test cross. So let's do the test cross. We are taking small b, smaller, smaller. Now we will cross these two. Let's see what will be the product. So from here we will get one type of gamete, that is small b and small r. Okay, and from here we will get 4 different types of gametes. Due to recombination, different types of combination we can get from here. Likewise, capital B with capital R. Okay, then capital B with small r, small b with capital R, and small b with small r. So these are the possibilities. So when we make crossings between these two, F1 and double recessive, we will get 4 different varieties of plants, 4 different products. So I can write here like this. These are the different gametes. We have this one. This is also a gamete from this parent. Now I can write it here, small b, small r. So what will be the different products? Let's write here. So when this capital B is small b, I can write here capital B, small b, capital R, small r. Likewise here, capital B, small b, smaller, smaller. Here small b, capital R, smaller. Here all are small, all are small. Okay, so focus on this. We have the first F2, first offspring, which is similar to one parent. So this is parental character. I am writing P here, parental. Okay, again look, this one is not found in any of its parent, so this is recombinant. I am writing R for this. This one is also recombinant, we can see here, because this one is also not similar to either of the parents. And finally this one is also parental, like the first one, because it is similar to this parent. Okay, so parental character along with some non-parental character or recombinant characters appears by the crossing between these 2 different plants. By test crossing the F1 generation, we can find 4 different offspring, out of which we have some parental and some recombinant characters. So why recombinant characters are appear here? Because here the genes are situated or located in different chromosomes, so they can combine to form different types of gametes. As we get recombinant gametes or different types of gametes, we can also find different types of offsprings, both parental as well as non-parental or recombinant. Okay, so now we will discuss the scenario 2. So what will be the scenario 2 when the genes are located in a single chromosome? Let's discuss. So this is all about your scenario 1, when the genes are located in 2 different or 2 separate chromosomes. Now let's see the scenario 2. I am writing here 2 because in this case we are going to talk about the genes located in the same chromosome. So genes, I am writing here genes on same chromosome. When the genes are present in the same chromosome, what will happen? So I will talk of a normal condition first. So here the condition is called cis arrangement. So cis arrangement means the dominant characters are present in homologous chromosome. So between the two homologous chromosome, one chromosome carries dominant character and the other one carries recessive character. So look how is the arrangement, cis arrangement. So it is RB and capital R, capital B. These genes are present in a single homologous chromosome. We know what is the structure of chromosome. So this is one chromatid and this is another chromatid. Okay. So these are two sister chromatids and together they form a single chromosome. So in this chromosome we have capital B here and here also, and capital R here and here. Okay, so this is the exact arrangement. I want to make you understand this. So another thing is that here all the dominant characters are there. This one is homologous to another chromosome. Okay, and that chromosome is having the small b and small r. So in this chromosome, I can draw it like this. In this chromosome, again, in this chromosome, I have small b here and small r here. Okay, clear? So these two are homologous chromosome. These two are sister chromatids of this chromosome and these are sister chromatids of this chromosome. Okay, and these two are non-sister chromatids. Clear? So these two are non-sister chromatids. Clear? So now look, we are going to crush these two types of flags. Okay, so here we have capital R and capital B, capital R and capital B. Here we have small r and small b. So by fusion of these two, we will get capital R, capital B, small r, small b. This is a normal condition. So normally this is found in the offspring. Clear? So when crossing over occurs, how the non-parental character appears, let's see. So what happens when crossing over occurs? So during crossing over, we have the two homologous chromosomes. Okay, so how non-parental character will found in the next generation? These two are located close to each other. So let me draw again this one, this chromatid and this one are located close to each other. Okay, so here the another one, they are located very close to each other. So here the crossing over occurs between the non-sister chromatids. Okay, so during crossing over, what will happen? Here we have capital B, we have capital B, and this one is our small b. This one is the small b. This one is the small b and this one is the capital B. So capital B is present in this chromosome and small b is in this chromosome. Clear? So when they separate from one another during meiosis, when they separate from one another, what happens? This, let me draw it with red so that you can easily understand. Okay, let me draw this chromosome in red color. So this one is the chromosome. Okay, so this one is the chromosome and crossing over occurs in this location. Okay, so due to crossing over, here we have the small b, here we have the capital B. So red color is for small b and black for the capital B. After crossing over, when they separate, the piece of non-sister chromatid between these two non-sister chromatid crossing over occurs. So when they separate, this red piece will come to the black chromosome and the black piece from the black chromosome comes to the red chromosome. So let's see what will happen when they separate. Okay, so look at this figure again. After their separation, they will be like this. This is one chromosome having the red part and small b, and this is another chromosome, and this one is having the black part with capital B. So the small b from this chromosome due to crossing over comes to this side, and capital B from here goes to that side. Okay, this is the result of crossing over. So all the other genes are in the same location. So I can write here, this one is your red, capital R, this one is capital R, and here it is the capital B. Okay, so here we have the smaller, here again we have the smaller, here it is the small b, but the small b from this chromosome is now in this chromosome, and the capital B from here travels to there. Okay, so this will result in a different condition. What will happen? Capital R, and the situation will be like this. This was the original situation. After crossing over, what will happen? This will become capital R, capital B, small b, and this will become smaller, capital B, small b. Okay, so this is the condition after crossing over. This will happen after crossing over. So this is the parental, this is parental character, this one is also parental gamete, but these two are recombinants. These two are recombinants. Clear? So these are the two recombinant variety. So now when we will go for test cross, we will have another plant having smaller, small b. So from here we have the gamete smaller, small b. Now what will be the result of the cross between these two? Let's see what will happen. Here capital R, capital B is there. So capital R and capital B is there. Again capital R, small b is there. Then small r, capital B is there. Then small r, small b is there. Okay, and it will fuse with smaller, small b. It will fuse with smaller, small b. I am writing here. This one is small r, small b. So what will be the result of the offsprings? Here we will have smaller, small b. Here we have smaller, capital B, small b. Here we have capital R, smaller, small b, small b. Here we have capital R, smaller, capital B, small b. So this is the result of the cross, of the test cross. Here this one is the parental character, and this one is also parental character, whereas these two are recombinant. Okay, so these two are the result of crossing over. Due to the crossing over between the non-sister chromatids of homologous chromosome, the resultant is having some non-parental characters which are called recombinants. Clear? So this is the scenario 2, when genes on same chromosome and both the homologous chromosome, one chromosome carries all the dominant characters and the other carries all the recessive characters, which is called the cis arrangement. Cis arrangement, and cis arrangement also is known as coupling. Coupling means the attraction between the dominant characters. The attraction between the dominant characters is known as coupling. They want to remain in a single chromosome where they are present. They want to be there. So dominant character want to be there in the chromosome, and recessive genes present in different chromosomes want to be there, and they want to retain their original position. So here, as the frequency of crossing over is very less, as the genes are located on the same chromosome, the frequency of crossing over is very less. So the parental characters, in this case, parental characters are seen in much higher percentage than recombinant or non-parental character. Okay, so the parental character here appears in much higher percentage as compared to the recombinant or non-parental character, because the genes are located on the same chromosome. Clear? So when they are located in different chromosomes, the percentage of recombinant will be more and that of parental character will be less. So now we will discuss about the trans arrangement. So this one is cis arrangement, when dominant characters are present in one chromosome and recessive are present in a different chromosome. But what will happen in trans arrangement? Let's see. I will draw the structure and show you. I am not erasing this one, the result of the cis arrangement, because it has a relationship with the trans arrangement. I will tell you at the end. So I have it in the board. So genes on same chromosome, but in this case we are telling it trans. Why it is called trans arrangement or repulsion? It is also called repulsion. So what is trans arrangement? Trans arrangement or repulsion? There the cis arrangement was coupling, because the dominant characters want to remain in one chromosome and the recessive genes want to remain in another chromosome. The tendency to remain together, the dominant, that may be dominant characters or dominant genes or recessive genes, the tendency to remain together is coupling, and here repulsion means they are present in different chromosomes. So let's see what is trans arrangement. In trans arrangement we have capital R. I am writing again with red marker. Capital R for the color, and small b for the smaller size of the flower. Likewise, smaller for white color of the flower, and capital B for big size of the flower. Okay, so now we are going to cross it with a double recessive. So first let's find out the gametes of these. So what will be the gamete? Here we can easily see why it is called trans arrangement. Yes, why it is called trans arrangement? Because here I will draw the structure of chromosome exactly. So here I have chromosome where there is capital R, small b. Okay. Likewise, another chromosome. In another chromosome or its homologous chromosome, we have the small r, capital B. Okay. So during crossing over, what will happen in this case? This B will shift to this side, the capital B will come to this chromosome, and the small b will jump to this side due to crossing over between these non-sister chromatids of homologous chromosome. The result will be capital R, the gamete from here, capital R, small b. Here it is capital R, this B, capital B is now in this chromosome due to crossing over. So I will write here capital B. Here it is smaller, and the small b is now here. So I will write small b, smaller, capital B. So now we will make fertilization between these different gametes and with the gamete of a double recessive parent for test cross. So we will have small r, small b to make the cross. Okay. So here we will see that these two are recombinant as a result of crossing over. These two varieties are the result of crossing over. So what will be the result? Let's discuss. So for in the trans arrangement, here I am writing it is about trans arrangement. In this case, what will be the result of the cross? Let's see. So here we have four different gametes. Let me write them. One is the capital R, small b, capital R, capital B, small r, small b, again here smaller and capital B. Okay. So the parent is having small r and capital B. So small r and capital B is the gamete with which we are going to make the cross. So what are the results? Let's write here. Capital R smaller, small b, capital R, capital B, small r, small b, smaller, capital B and small b. So these are the different products we will get from this. So here we have capital R smaller, small b. This one is parental. I am writing P for it. Again, capital R smaller, capital B, small b. Smaller, capital B, small b. This one is also parental. Why? Because this gamete is parental. This gamete is parental and this gamete is parental. So due to the fusion of this gamete and this one is formed, so it is parental. Again, by the fusion of this gamete with the double recessive one will result into parental combination, whereas the two at the middle results in the formation of recombinants. Okay, so these two are non-parental. So as a result of the cross, we will have two recombinant variety and two parental variety. Okay, so I have the result of cis already here, and now we have the result of trans. So let's compare the result between the trans and cis arrangement of the genes in the chromosome. So here the recombinants, look at the recombinants first. Capital R smaller, capital B, small b. Capital R smaller, capital B, small b. Here the first recombinant is here in case of cis it is parental. Next, let's see this one, smaller, small b. Here it is also parental. So the parental characters in the cis arrangement, in the result of cis arrangement, is equal to the recombinant in the trans, and the parental in the trans are equal to the recombinant in the cis. Look, this one is parental, capital R smaller, both are small, capital R smaller, both are small b. Here smaller, capital B, small b, smaller, capital B, small b. So these two are recombinant in cis, whereas parental in trans arrangement, and these two are recombinant in trans arrangement, whereas they are parental in the cis arrangement. So this is the similarities, this is the point we need to note that parental characters in cis arrangement are recombinant in trans and parental in trans are recombinant in cis arrangement. Okay, so remember one thing, the genes in different chromosomes, when the genes are present in two different chromosomes, the chance of crossing over is more, so the recombinant will be more. But here, as the genes in same chromosome, so the parental characters are appear in higher percentage, much more higher than the non-parental or recombinant varieties. So this is all about the condition, the scenarios. The first one is when the genes are located in different chromosomes, how the appearance of recombinant variety is more. In scenario 2, we have discussed about two different things, the arrangement of genes, the dominant one side in one chromosome and recessive on the other chromosome. In that case, we have the cis arrangement and trans arrangement. In cis arrangement, we have all dominant characters in one chromosome and all recessive in the other. Whereas in trans arrangement, we have two dominant, here the red color but size is small, here the white color but the size is larger. So one character is dominant, one is recessive, here also one is dominant, one is recessive. So this type of arrangement is the trans arrangement. So this is all about the linkage and what is the result of crossing over. So these are the different conditions we will study in linkage and crossing over, and these crossing over results into appearance of recombinant variety in the offspring. So this is all about the concept and detail about the linkage and crossing over. Thank you very much.